Hi Year 9, this is the first part of our biology material. So this is B1. Again, I'm going to video it in two different parts just to make it slightly shorter each time. Hopefully this first video is just going to build on what you already know from your previous topics in Year 7, 8 and 9, so it's fairly straightforward. We're going to look at structure of the plant cells and animal cells and compare them. We're going to look at the roles of different organelles and that sort of thing. And then the second part of the video is a little bit more challenging. We're starting with diffusion and we're going on to osmosis. We're going to look at active transport and also movement and exchange of materials. So hopefully this first part of the video is just cementing those ideas that are already in place for you, and then the second part will be building upon that again. Here we go, much better than me trying to remember everything that I'm going to go through. Here's a quick breakdown of what's going to be in both parts of the video for Biology B1. The first part of this chapter is looking at microscopes. The development of the microscope is really important because prior to this we didn't have any understanding about cells, we didn't know about bacteria. People used to think that animals came from inanimate objects. So for example, they used to think that because mice came out of barns, that the mice was not born to but came from the barn. They didn't have an idea about gestation and that kind of thing. We also thought that disease came from bad smells. So by using a microscope they were able to see bacteria and had a better understanding about that kind of thing. A little bit later in the video we're going to look at a required practical that you guys need to understand and be able to do that's linked to microscopes. But just for now, have a look at the different labels that I've put on this microscope, as they may ask you to label each part of the microscope. The picture I've used here is of a light microscope, so it uses a beam of light to form an image and it can magnify up to 2,000 times. So these are the ones you'll see in school. However, in the 1930s they invented something called an electron microscope. This allowed biologists to see and understand more about the structures inside the cell. We call the structures subcellular structures. Electron microscopes are often used because they can magnify at 1,000 times stronger than a light microscope. So some of them can also do 3D imaging. But obviously and unsurprisingly they're much more expensive, and because of the equipment used in them they have to be held at special temperature and pressure and even humidity controlled rooms, so no surprise we don't have one of those at school. So moving on from this, we could actually use the information we collect from the microscope to calculate the size of one of the subcellular structures or even a whole cell that we see down the microscope. In order to do this, we need 2 out of 3 of the pieces of information you see in the triangle here. So we've got image size, which is what you're looking at. So sometimes when we're working with microscopes at school, students will say to me, "Oh miss, I can see a ruler down the microscope." But in actual fact, that's there to help you work out your image size. The next one is actual image. So that's the true size of the cell that you're looking at. And the last one is the magnification. So you'll be able to see this on the lens you're using on the microscope. For this calculation, you can rearrange the equation using the triangle method. I remember this triangle with I am, just as you've seen it laid out here, but actually it doesn't really matter which way the A and the M go because the line between them is the multiplication, so really it can be either way round. In an exam, this question could be given to you any way round, so they could give you, say, the actual image and the magnification and ask what the image size would be, but really when you're working in a lab, the most likely case scenario is that you'll get an image size and you'll know your magnification and you'll be trying to work out the actual image. So let's use that as an example now. So the first thing you'll need to do is work out your own magnification. You can do this by multiplying the magnification of your eyepiece lens by the magnification of your objective lens. In our example, I've used a 4 magnification and a 10 magnification, giving me an overall magnification of 40. The next thing you'll need to do is to take a measurement of the image you're seeing. So in our example, it's 3.12 millimeters. So the final thing you need to do is put all of this information into our equation. So to calculate actual image, we need to divide the image size by the magnification used. So in our example, we've got 3.12 millimeters divided by a magnification of 40. This gives us 0.0078 millimetres, or this can be simplified to 78 micrometres. In order to put your answer into micrometres, you need to know the conversion rate between millimetres and micrometres. So 1 millimetre is equal to 1,000 micrometres. Hopefully that has covered your understanding of magnification using a microscope. So quite simply, magnification is making something look bigger. However, another thing they make talk about when they're comparing microscopes is a resolving power, or the microscope's resolution. So the resolving power of a microscope is the microscope's ability to distinguish things as two separate items. We could think of this as how clear an image might look. So the more powerful microscopes are able to give us a clear image of objects that are really close together. A microscope with a lower resolving power would need a greater distance between objects to give us a clear image. I'm really aware that we've spent a long time talking about the microscopy part of this topic, so I'm just popping this here. This is your required practical breakdown. So you'd be expected to be able to make a slide of a plant or animal cell and observe it underneath a microscope. So you can look at the details here, but I'm not going to go over it because you're going to do this in class when we get back to school. The next part of this topic is comparing animal and plant cells. This should be something you're fairly familiar with as you've gone through this in your Key Stage 3 topic. So here are just two images, one of an animal cell and one of a plant cell. So when comparing plant and animal cells, you need to know which organelles are found in animal cells and plant cells. I find it much easier to compare these by using a Venn diagram. This shows you which organelles are found in both plant and animal cells and which are just found in plant. You should notice that there are no organelles that are found only in animal cells. Each organelle is unique and has its own role within the cell. Again, you've looked at these in Key Stage 3, but we'll run through them really quickly. So the nucleus controls the activity in the cell and contains the DNA. The cytoplasm is the liquid section, and this is where chemical reaction occurs. The cell membrane controls movement in and out of the cell. We have a cellulose cell wall. It's important that you use the word cellulose because not all cell walls are made of cellulose. So this strengthens and supports the cell. Mitochondria is where aerobic respiration takes place. Ribosomes are where proteins are made. Chloroplasts are where photosynthesis occurs, and this is also where you'd find chlorophyll, and that's what makes it green. We've also got a permanent vacuole within plant cells. This is filled with sap, and it gives the plant cells a turgor pressure, which makes them feel rigid and provides support to the plants. Okay, so that's the end of that section. I've just whipped through it really quickly because none of it is new information to you. We now need to compare eukaryotes and prokaryotes. A eukaryotic cell, I always remember, is a plant or animal cell because I always think eukaryotic as in you, like you the person. So maybe that's helpful to you, I don't know. And prokaryotic cells are looking at bacteria cells. So if you know me, if you've worked with me before, you know that I really like a table. So I've put together this quick comparison of the two so you can see how they're similar and how they're different. So the first comparison point, we're looking at what is an example of a eukaryote and prokaryote. So a eukaryote could be an animal, plant, fungal or protist cell. Generally, these are multicellular organisms. This means that it's an organism made up of many specialised cells, all needed to enable the organism to function. Prokaryotes are bacterial cells, and these are unicellular organisms. This means they're made up of one cell and are able to survive with their one cell carrying out all the functions needed. So I've also put in this section that eukaryotes are larger than prokaryotes. We're going to talk about orders of magnitude. One order of magnitude means that an object or a cell or an organism is 10 times larger than the other. So a prokaryote is 1 to 2 orders smaller than a eukaryote. This means it's 10 to 20 times smaller than a eukaryote. Orders of magnitude can be expressed using powers of 10. So if a eukaryote was 2 orders larger than a prokaryote, you could say it was 10 to the power 2, bigger than the prokaryote. So next you'd expect to find cytoplasm and cell membranes in both eukaryotes and prokaryotes. Not all eukaryotes have a cell wall. For example, we know that plants do, but animals do not. Bacterial cells do have a cell wall, however it's not made of cellulose. Some also will have a protective sign capsule that you'd find just outside of the cell wall. We can also look at how the DNA is different. So in a eukaryote, the genetic material will be found within a nucleus. However, with our prokaryotes, there is no nucleus present. The genetic material is found loose in the cytoplasm, but they also have circular plasmids of DNA within their cytoplasm. And finally, prokaryotes have long flagella or cilia to help them to move. The final thing we need to talk about in part 1 of B1 is specialised cells. So cells become specialised within animal and plant cells. They originate as something that we can refer to as stem cells, and after this they become specialised for a specific function. In both plant and animal cells, they differentiate to become specialised at their job. This means that they're going to have specific adaptations that make them good at what we need them to do. Firstly, let's look at animal cells. Here is an example of a nerve cell. You may have seen this in your Key Stage 3 topics, but you won't have looked at this in detail. You will be expected to know the labels on this cell, and you'll also be expected to know how it's adapted for its function. So the job of a nerve cell is to send impulses from one area of the body to another. Here are the adaptations. It has many dendrites. This enables it to make connections with other cells. This means we're able to send impulses from one area to another relatively quickly. Having long axons also helps us to send impulses quickly. This is because we do not need to pass an impulse across a synapse if we're able to send the impulse straight down the axon. Incredibly, the longest axon you'd find in a blue whale can be up to 25 meters long. However, in a human, our longest axon runs from our spine to our big toes. Next, the nerve cell has synapses. These are small gaps between one nerve cell and the next. This passes an impulse along using chemicals. Finally, you'd find a lot of mitochondria in the cell body of a nerve cell. This provides plentiful energy to make transmitter chemicals. Next, we will look at muscle cells. The role of a muscle cell is to relax and contract to enable movement in animals. Striated muscles have the following adaptations. Firstly, they contain special proteins that slide over one another, allowing the fibres to contract. They also contain many mitochondria to transfer the energy needed for the chemical reactions to take place as the cells contract and relax. And finally, they can store glycogen. This is a chemical that can be broken down and used in cellular respiration by the mitochondria to transfer energy needed for the fibres to contract. The last specialised animal cell you need to know about is a sperm cell. The role of the sperm cell is to deliver the male's DNA to the egg. They have the following adaptations. Sperm have long tails which whip from side to side, which help them to move to the female reproductive system. They also have a middle section, and this is full of mitochondria. This transfers energy that's needed for the tail to do its work. There is an acrosome in the front part of the sperm, and this stores digestive enzymes which breaks down the outer layer of the egg, allowing the sperm to get in. And finally, it has a large nucleus to contain all of the genetic information that needs to be passed on. So that's everything you need to know about the animal cells. Now we're going to look at how plant cells have been specialised in order to carry out specific functions. Firstly, we're going to consider root hair cells. The role of the root hair cell is to help take up water and mineral ions into the plant. In order to do this job effectively, they've been adapted in the following ways. Firstly, root hair cells have a high surface area. This increases the movement of water into the cells. They also have a large permanent vacuole, and this speeds up movement of water by osmosis from the soil into the root hair cell. We'll look at osmosis a little later. And finally, they have many mitochondria that transfer the energy needed for active transport of minerals into the root hair cells. Again, part 2 we'll look at active transport. The next specialised plant cell we're going to look at are photosynthetic cells. The main role of photosynthetic cells is to take in light and make food via photosynthesis for the rest of the plant. The cells are specialised because they contain green structures called chloroplasts, and these contain chlorophyll. This traps the light needed for photosynthesis. They are also usually positioned in continuous layers in the leaves and in the outer layers of the stem. This means they can absorb as much light as possible. finally, they have a large permanent vacuole that helps keep the cell rigid. When lots of these rigid cells are arranged together to form photosynthetic tissue, they help to support the stem. They also keep the leaf spread out so it can capture as much light as possible. The third specialised plant cell we're going to look at are the xylem cells. These cells come together to form a transport tissue in plants. It carries water and mineral ions from the roots to the highest leaves and shoots. There are two main ways that xylem cells are adapted to their function. Xylem cells are alive when they're first formed, but a special chemical called lignan builds up in spirals in the walls. The cells die and form long hollow tubes that allow water and minerals to move easily through them from one end of the plant to the other. The spirals and rings of lignan in the xylem cells make them very strong and help them withstand the pressure of water moving up the plant, and they also support the plant stem. The last specialised plant cell, and last part of this topic, is looking at phloem cells. These are cells that transport food made by photosynthesis around the body of the plant. They are specialised in two specific ways. Firstly, the cell walls between the cells break down to form something called sieve plates. These allow water carrying dissolved food to move freely up and down the tubes to where it's needed. Secondly, and finally, phloem cells lose a lot of their internal structures, but they're supported by companion cells that you'll see just outside of the phloem cells. These help to keep them alive. The mitochondria of the companion cells transfer the energy needed to move dissolved food up and down the plant. So that was part 1 of B1. Please look out for part 2 of B1 to finish the topic.